Welcome to lecture 2 on metabolism, metabolic pathways for carbohydrates. So next let's discuss the next pathway, which is the pentose phosphate pathway. So this is an alternative pathway for the oxidation of glucose. So it results in the production of the coenzyme NADPH and the 5-carbon pentoses. So NADPH is a coenzyme that is the reduced form of NADP plus, is a very important coenzyme, and it's required in anabolic pathways including the biosynthesis of nucleic acids, cholesterol and fatty acids in the liver and fat cells. So let's take a look at this pathway here. So the pentose pathway begins with the glucose 6-phosphate, so the same molecule that you start with glucose as well in the from the reaction 1 of glycolysis. So which is then converted into ribulose 5-phosphate and requires 2 NADP plus molecules. And this ribulose 5-phosphate is then isomerized by the catalyst phosphopentose isomerase to become ribose 5-phosphate. And an important component of the nucleotides, which is going to be useful to create ATP, GTP, UTP, NAD+, FAD, and RNA. So all of these are molecules that can be created by just this one molecule, ribose 5-phosphate. So in summary, you have glucose 6-phosphate combining with 2 NADP plus molecules and water, forming ribose 5-phosphate with 2 NADPH molecules with 2H plus CO2. So this involves a series of reactions where 3 ribose 5-phosphate molecules are then converted into 2 hexose molecules and 1 triose molecule. So what happens here is that the 3 ribose 5-phosphate molecules are converted into 2 fructose 6-phosphates, and then you get glyceraldehyde 3-phosphate. Now, the pentose pathway converts basically glucose 6-phosphate into ribose 5-phosphate, which is generally one that is needed for the synthesis of both DNA and RNA. And this ribose 5-phosphate can also return to glycolysis as needed, so which can end up becoming fructose 6-phosphate, which can convert to glyceraldehyde 3-phosphate and undergo glycolysis again. Remember that this is a double-sided pathway, which means that it can get out of the glycolysis and it can enter into the glycolysis at any point in time. So the pathways for this pyruvate, let's talk about the pyruvate molecule and what are the pathways. Now we said that the lactate is generally the material that is generally transported to the liver, which is there converted back into pyruvate. Now whenever you do rigorous exercise and rapid breathing, it helps to repay the oxygen debt incurred during the exercise. Let's talk about the pathways for pyruvate. So the pyruvate here produced from glucose can now enter into the pathways that continue to extract energy. Now the available pathway depends on whether there is a sufficient oxygen in the cell. There are 2 types of conditions that we are thinking about. One is aerobic conditions, the other is anaerobic conditions. Aerobic is where there is enough oxygen, and anaerobic is then where there is no oxygen present or not enough oxygen present. Now what happens when you have aerobic conditions? So the oxygen is available to convert the pyruvate into acetyl coenzyme A and carbon dioxide. Now when the oxygen levels are really low, pyruvate ends up being reduced to lactate. So that's this lactate is the reason why you have muscle cramps, muscle pain. So under anaerobic conditions, what happens here? The pyruvate moves from the cytosol into the mitochondria to be oxidized further. Now undergoes oxidation when you have a carbon atom is removed as carbon dioxide and the coenzyme NAD plus is reduced to NADH, and the 2 carbon acetyl groups is attached to the CoA, producing acetyl CoA, which is an important intermediate in the many metabolic pathways. So you have pyruvate mixing with CoA and NAD plus under the use of pyruvate dehydrogenase to create acetyl CoA, carbon dioxide, and NAD plus reduced to NADH. Under anaerobic conditions, what happens here is that when we engage in exercise, rigorous exercise, so the oxygen that is in our muscles generally gets depleted, and whenever you start breathing rapidly, you start to repay the oxygen debt that you are incurring. And this anaerobic conditions where there is no oxygen, so the pyruvate is generally reduced to lactate and NAD plus by lactate dehydrogenase. So this NAD plus is then used to oxidize the glycerol 3-phosphate in the glycolysis pathway and ends up producing a small amount of ATP. So you start with the pyruvate. Notice that there is no oxygen here. So NADH combined with NADH, so you use the enzyme lactate dehydrogenase to create the molecule lactate and NAD plus. So this here in pyruvate, when you have anaerobic conditions, the accumulation of this lactate can cause muscles to tire and become sore. So the lactase is generally subsequently transported to the liver and then converted back to pyruvate. But under anaerobic conditions, the only ATP production in the glycolysis occurs during the steps that phosphorylate ADP directly, giving a net gain of only 2 ATP. This is the reason why as you exercise more and more you get tired and your muscles become sore. The reason mainly is because there is not enough energy, or there is not enough oxygen to produce more ATP, and you have less ATP production. The lesser the ATP production, the lower the endurance of your own muscles. So you have glucose with 2 ADP and 2 phosphate creating 2 lactate molecules and 2 ATP molecules. So there is not enough ATP created. That is the reason why anaerobic conditions are something that are different from aerobic conditions. So this is how anaerobic and aerobic conditions differ. Now anaerobic conditions, there are 2 types. In the skeletal muscle, it creates a lactate. Under fermentation processes, this is also an anaerobic condition where it creates acetaldehyde and carbon dioxide. The acetaldehyde, under the use of NADH enzyme, finally undergoes the formation of ethanol. So anaerobic conditions both are the same, but in your cells it creates lactate, which is then transported to the liver to convert back into pyruvate. But here in terms of the fermentation, when you use yeast, it creates acetaldehyde. The acetaldehyde further undergoes further oxidation to create the formation of, further reduction to create ethanol. So let's talk the fermentation. So some microorganisms, particularly yeast, can convert sugars to ethanol by fermentation. So this fermentation occurs in anaerobic microorganisms such as yeast, and it generally undergoes decarboxylation of the pyruvate to create acetaldehyde, which is then undergoing reduction to create ethanol, regenerating the NAD+ molecule. So you have pyruvate, under pyruvate decarboxylase creating ethanol and carbon dioxide. The ethanol, which is the acetaldehyde, combines with NADH to create ethanol, which is generally the final compound that you use under the use of the enzyme alcohol dehydrogenase. So during fermentation, what is happening here is that the enzymes in the yeast are basically converting sugars to glucose, and then those glucose into ethanol and carbon dioxide gas. So the fermentation, when you use barley malt, it produces beer. When you use sugars, it produces wine and champagne. When you use potatoes or grain, it produces vodka. When you use rice, it produces sake. When you use corn or rye, it produces whiskeys. So this is the type of material that you need to produce a particular product of ethanol. So pause the video right here and try to answer these questions. Next, let's talk about glycogen synthesis and degradation. So glycogen here, one of the problems within your body is that you can store energy in the form of glycogen. So glycogen storage diseases occur when you have a defective enzyme that is involved in the pathway of glycogen storage or degradation. So this is a micrograph that shows the excess amount of stored glycogen in a liver biopsy of a patient with Cori's disease. So let's now talk about, the goal here is to understand the synthesis and breakdown of glycogen. So what is glycogen here? Whenever you have excess glucose in your blood, excess glucose is generally used to replenish the energy reserves and synthesizing glycogen, so which is then stored in your muscles and liver. So this glycogen is a polymer of glucose with 1,4 glycosidic bonds and multiple branches attached by alpha-1,6 glycosidic bonds. And it is generally formed when you have high levels of glucose 6-phosphate in the first reaction of glycolysis. And this glycogen is not formed when energy stores are full, which means that additional glucose is generally converted to triacylglycerols and stored in your body as fat. Remember that there is only some amount that can be stored as glycogen. The rest of it is stored in the form of triacylglycerols or TAGs, and which generally forms body fat. Next, let's talk about glycogenesis. Glycogenesis is the metabolic process of converting glucose molecules into glycogen. So this produces glucose 6-phosphate in the reaction 1 of glycolysis. So what is the reaction 1 here? You have glucose 6-phosphate under the use of the enzyme phosphoglucomutase. It creates glucose 1-phosphate and uridine using the enzyme phosphoglucomutase. Now this glucose 1-phosphate under the use of the enzyme pyrophosphorylase undergoes the formation of UDP glycose. Now this reaction here is, there is an energy release when you have pyrophosphorylase catalyzing this reaction. The high energy compound UTP transfers UMP to glucose 1-phosphate to give UDP glucose and pyrophosphate PPA. Now the last production where you have glycogen synthesis. So glycogen synthase is the enzyme that we end up using, where the UDP glucose undergoes glycogen chain formation, and it is either, there are two ways you can regulate this. One, it's activated by insulin and it is inhibited by glucagon. So glucagon and epinephrine. So glucagon if it's in the liver and epinephrine if it's in the muscle. Now this is one of the most important processes that happens in your body, and this is one of the most important processes that breaks down, that is used in the storage of energy in your body. Excess glucose is always stored in one form or the other. Patients with diabetes do not have the enough levels of insulin, which means that there is excess blood glucose in their blood which is not converted to glycogen, which then creates problems in your body. So not enough glycogen production, not enough glycogen conversion is a big problem in your body. Next, let's talk about glycogenolysis, which is reactions 1 and 2. So in glycogenolysis, you have glycogen which is broken down to glucose, and in reaction 1 you have phosphorylasis, where the glucose molecules are removed from the glycogen chain and the glucose molecules are phosphorylated by glycogen phosphorylase to yield glucose 1 phosphate. And in the reaction 2 where you undergo hydrolysis, where the alpha 1,6 glycogen phosphorylase cleaves the alpha 1,4 links until one of the glucose remains bonded to the main chain, and you have a deep branching enzyme that breaks up these bonds to end up creating the hydrolyzed reaction from glu1. So you have the glycogen that is broken up into two parts. One, it's into broken up each molecule into glucose 1 phosphate, which ends up creating glucose with 1 glucose removed, and the enzyme that is used is glycogen phosphorylase. It is activated by glucagon and epinephrine, and it is inhibited by insulin. Now in reaction 3, what's going to happen? You have isomerization of the glucose 1 phosphate molecules to convert it to form glucose 6 phosphate, which then enters into the glycolysis pathway at reaction 2. Remember that all of these are happening inside of a chain of reactions that are helping one another. The main process of production of energy is glycolysis, but the products that you end up with storing can end up coming back to the pathway at some point inside the pathway. So in reaction 4, you undergo dephosphorylation, where the cells in the liver and kidneys have a glucose 6 phosphatase that hydrolyze the glucose 6 phosphate to end up creating free glucose. So the free glucose then enters into the glycolysis process as well. So this is the idea behind the process. So pause the video and try to answer these questions. Next, let's talk about the regulation of glycogen metabolism. So the brain, skeletal muscle and red blood cells require large amounts of glucose to function properly. So to protect the brain, hormones with opposing actions control the blood sugar levels, such as 3 of them: glucagon, insulin and epinephrine. So let's talk about the chemistry that is linked to some of the health diseases that end up due to the cause of glycogen storage diseases. So these are diseases that can occur when you have a defective enzyme that is involved in the pathway of glycogen storage or degradation. And you have severe glycogen storage diseases that are detected in infancy, and you're going to expect that the life expectancy is usually very short, and less severe types are generally detected in adulthood. So one of the common ones that you will end up seeing is the disease glycogen synthase deficiency, which can cause liver type severe and lack of muscle glycogen and exercise intolerant people. You have von Gog's disease, which can not convert the glucose 6 phosphate to glucose, which ends up creating glycogen accumulation, liver enlargement, hypoglycemic behavior and short life expectancy. So when you have the lysosomes of all kidneys where you have the defective enzyme alpha 1 to 4 phosphatase, you can have Pompe's disease. Pompe's disease where you have glycogen accumulation in the heart, and it results in heart failure, significant muscle weakness and a short life expectancy. In liver and muscle, you have the enzyme glycogen debranching enzyme, which can cause Cori's disease, which cannot completely degrade the glycogen to glucose, which can create lack of glycogen degradation, which can hydrolyze, so they cannot hydrolyze the alpha 1 to 6 glycosidic bonds, end up creating hypoglycemic problems and muscle weakness. In the liver, you also have the same enzyme, which can create Anderson's disease, which cannot form the alpha 1 to 6 glycogen branches, which can create abnormal specifics, and you have unbranched glycogen accumulation, results in short life expectancy due to heart or liver failure. In the muscle, you have glycogen phosphorylase, which can cause McArdle's disease, which cannot degrade the glycogen in the muscle to glucose, which can create exercise intolerance, muscle cramps due to exercise. And you have finally liver, you have glycogen phosphorylase, which can cause Hertz disease, which cannot degrade glycogen to glucose in the liver, which generally has very mild impact. So next, let's talk about gluconeogenesis, which is the process of glucosynthesis. So glucose is synthesized in the tissues of the liver and kidneys. Remember this mainly. There are two processes where you have two main organs. These tissues are that use glucose as the main energy sources, that are brain, skeletal muscle, and red blood cells. These are the three that are going to use. Now glucose is the primary energy source for the brain, skeletal muscle, and red blood cells, and any deficiency of this glucose can impair brain and nervous system function. Now if our glycogen stores are depleted, what will happen is that liver start to synthesize glucose by gluconeogenesis. So where the glucose is synthesized in the cytosol of the liver cells and some is synthesized in the kidneys. How are they synthesized? So glycogen can supply us with about 1 day's requirement of glucose, and it's generally made up of glucose, so most of which is synthesized in the cytosol of the liver cells. So now to begin gluconeogenesis, the carbon atoms from the non-carbohydrate food sources are generally converted to pyruvate. So to start the synthesis of glucose from pyruvate, so you have two catalyzed reactions that are needed to replace reaction 10 in glycolysis, where you have pyruvate carboxylase uses the energy of ATP hydrolysis to catalyze the addition of CO2 to pyruvate and produces oxaloacetate. You have phosphoenol pyruvate carboxygenase, which can convert oxaloacetate to phosphoenol pyruvate. These molecules now use the enzymes to finally convert it to form fructose 1,6 biphosphate. So you have a pyruvate molecule which undergoes under presence of carbon dioxide, ATP, and H2O, undergoes pyruvate carboxylase, where it adds the carbon dioxide to create oxaloacetate. The oxaloacetate, which then undergoes the use of GTP under the enzyme phosphoenol pyruvate carboxygenase, converts oxaloacetate into phosphoenol pyruvate and releases carbon dioxide and GDP. Now this fructose 1,6 biphosphate is then converted to fructose 6 phosphate, which is the second irreversible reaction in glycolysis, is generally bypassed by the fructose 1,6 biphosphatase, which is generally going to cleave the phosphate group from fructose 1,6 biphosphate. So the product fructose 6 phosphate undergoes the reversible reaction 2 of glycolysis to yield fructose 6 phosphate, which uses the enzyme glucose 6 phosphatase, where the glucose 6 phosphate is generally converted finally into free glucose, which ends up going back into the bloodstream. Now glycolysis and gluconeogenesis are reverse processes of each other, but they enter the bloodstream in different ways. Remember that here in glycolysis we are breaking down glucose to create pyruvate. In gluconeogenesis we are using pyruvate to create free glucose. So this is the idea behind glycolysis and gluconeogenesis. Now what is the energy cost of gluconeogenesis is that the pathway consists of 7 irreversible reactions of glycolysis and 4 new reactions that replace the 3 irreversible reactions. Overall, glucose synthesis requires 4 ATPs, 2 GTPs, and 2 NADHs. So next let's talk about the lactate and the Cori cycle. So the Cori cycle is basically the flow of lactate and glucose between the muscles and the liver. So it occurs when you have anaerobic conditions that occur in the active muscle, and the glycolysis produces lactate. So this happens when when lactate moves through the bloodstream to the liver, where it is oxidized back to pyruvate. So where it converts the pyruvate back to glucose, which is then carried back to the muscle. So remember that lactate is something that cannot be broken down to glucose directly by the muscle under anaerobic conditions. So that's the reason why you need the liver as the glucose synthesizer for the cycle. So what happens in glycolysis? You have glycogen, which is converted to glucose to pyruvate. Pyruvate is converted into lactate. Lactate then goes into the bloodstream and enters the liver. In the liver, it becomes lactate, becomes pyruvate again. Pyruvate is then converted into glucose and stored in the form of glycogen, which then enters back into the bloodstream and enters back into the muscle for it to be working back again. So this here is the this is the Cori cycle. So how do you regulate the gluconeogenesis? Glucogenesis is not utilized when the diet is high in carbohydrates. This process is very active when the diet is very low in carbohydrates. This is very important because when your body enters gluconeogenesis, it can convert other cells into glucose. So when conditions in the cell favor glycolysis, there is no synthesis of glucose. When the cell requires the synthesis of glucose, glycolysis is turned off. Remember that one process is the opposite of other, which means that if you want glucose to form, it ends up creating glycolysis. So when you want synthesis of glucose, you undergo your body undergoes gluconeogenesis and glycolysis is switched off. When your body is already high of carbohydrates, then it undergoes glycolysis and forms, and glycolysis occurs and gluconeogenesis is turned off. So glycolysis is activated by high glucose levels and insulin, and gluconeogenesis is activated by low glucose levels and glucose 6-phosphate. And it is inhibited by glucose 6-phosphate and epinephrine. And the enzyme phosphofructokinase, in terms of the glycolysis, it is activated by AMP and insulin, is inhibited by ATP and glucagon. In the enzyme pyruvate kinase, in terms of gluconeogenesis, for creation of fructose 1,6 biphosphate, you have the activated by low glucose levels and glucagon, inhibited by AMP and insulin. The enzyme pyruvate carboxylase or pyruvate kinase is activated by fructose 1,6 biphosphate, is inhibited by ATP and acetyl-CoA. The enzyme pyruvate carboxylase is activated by low glucose levels and glucagon and inhibited by insulin. So this is the entire metabolic process where you have polysaccharides are converted into glucose under digestion. In the pentose pathway, it creates ribose and NADPH. In the glycolysis, it can convert it into pyruvate. Pyruvate then converts into Cori cycle into lactate, other processes into acetyl-CoA, and in fermentation into ethanol. In the reverse, it undergoes gluconeogenesis, where the pyruvate converts back into create glucose. In glycogenolysis, it can create glycogen. In it can glycogenolysis, glycogen is converted to glucose. In glycogenesis, glucose is converted into glycogen to store the energy in the form of glycogen. So pause the video right here and try to answer these questions. So this is the concept map of everything that we discussed today. So we have discussed all the pathways for the digestion of carbohydrates or the metabolism of carbohydrates in your body. Glycolysis, glycogenolysis, glycogenesis, pentose pathway, Cori cycle, fermentation, and we also talked about the process of glycolysis and gluconeogenesis and each of their inhibition and activation. So with this, we end our lecture on metabolic pathways of carbohydrates.